Okay, everyone. Thank you so much and welcome for joining our webinar series at the Center for Sustainable Tropical Fisheries and Aquaculture here at James Cook University. I'm very happy to introduce you today to our speaker, Professor Cynthia Reginus from University of Queensland. Professor Cynthia is an evolutionary geneticist with wide-ranging interests, spanning biogeography, phylogeography, molecular ecology, population genomics, speciation, hybridization, invasive species, and conservation. Overall, her research seeks to understand how marine biodiversity is created, where that biodiversity has accumulated, and how this knowledge can be used to preserve biodiversity and the processes that create it in a changing world. She's especially fond of mollusks, reef fishes, and corals, but easily distracted by other taxa as well. Cynthia has been at University of Queensland since 2006 and previously held an endowed postdoctoral fellowship in molecular evolution and comparative genomics at Duke University. So before I hand over to Cynthia, I just want to remind you, everyone, that if you have any questions during the presentation, please place them in this Q&A option that you can see at the bottom of your screen, as there will be some time for discussion at the end of the presentation. And with further ado, Cynthia, thank you so much for joining us, and I'll hand it over to you now.
All right, well, thank you, Katarina, for organizing this, and thank you for coming today. And I'm really excited to talk about this topic area. When I was a postdoc, which now seems like a really long time ago, I spent a lot of time thinking about molecular evolution and hybridization in marine mussels. And I knew when I came to Australia 14 years ago that there's going to be some really interesting stories to tell with Australian mussels. But like so many things, sometimes it takes a long time to get around to some of those interesting bits, and it's taken a long time to get funding to work in this system. But that's okay, because in the meantime, genomics has really changed the way we do biology. And so there are questions that we can ask now about mussels and other creatures that we really couldn't ask 14 years ago. So in some ways it was worth waiting, and I hope that you will agree. So I want to start this talk in a slightly philosophical manner, and to think about when is it that we value genetic diversity. So, sorry, I went for, oh dear, it's not going backwards. Anyways, on the previous slide, there were pictures of iconic Australian marine species, and I think we would all agree that those species are intrinsically valuable, partially because they're so phylogenetically distinctive. But when we consider organisms that are more closely related to each other, such as dingoes and dogs, which diverged 4,000 to 6,000 years ago, it gets a little more tricky to say when do we value that endemic genetic diversity. And so I'm just going to use dingoes as just sort of a parallel to set us up in thinking about how we might approach endemic diversity. So dingoes are a recent species in Australia. And if we want to use genetics to distinguish between dingoes and dogs, we run into problems very quickly. So this is a phylogenetic tree and then a haplotype network, which might be familiar to many of you, where each circle represents a distinct mitochondrial haplotype. And so the main take-home message here is that this most common haplotype is shared between dingoes and a dog. And so if you were to pull out a sequence of mitochondrial DNA and you end up with this haplotype, you wouldn't know whether the individual you'd sequenced was really a dingo or a dog. And this is similarly tricky with the Y chromosome. Just another example that although there is a somewhat distinctive chromosome here in Australia, the New Guinea singing dogs share that chromosome. So again, if you pulled out a sequence of Y chromosome, you wouldn't know whether you had a dingo or a dog in terms of that individual. And so this speaks to the fact that these two taxa are very recently diverged from each other. And so that recent divergence makes it quite tricky for distinguishing between species. And this gets worse when we have hybridization going on, because this will, of course, intermix alleles that originate from different taxa. And so, you know, we can imagine that with thinking about dingoes and dogs, that there are many possible outcomes. Hybridization happens between them, we know that. Undoubtedly, there's gene introgression going on between them. If there's a lot of hybridization, they might become a hybrid swarm, which eventually could lead to genetic extinction, or maybe enhanced invasive spread using combinations of alleles from different species. And so this gives us a backdrop of some of the things that can happen when we have hybridization going on between a native taxon and a non-native taxon. So now I want to turn to organisms that I personally find much more exciting than dingoes and dogs, although I'd be very excited to see a wild dingo, I will confess. So I want to think about Mytilus blue mussels for a minute. And so what we're looking at here is a bed of mussels, and we actually have two different species and hybrids in this picture. And I certainly cannot tell who's who by looking at them. And it turns out that we need genetics in order to be able to tell them apart. So blue mussels have very plastic morphology, and the shells are not particularly good for telling species apart. And of course, blue mussels are really important aquaculture species. For example, in Europe, they're the fourth largest group that is farmed by tonnage. And in Australia, the blue mussel farming industry is worth $12 million and growing. And I think really importantly, mussels are an excellent source of protein in the sense that their propagation is very low impact, it's very clean, and then the meat itself is high in protein, low in fat, very healthy. And so, you know, we should be farming mussels and we should be eating them, because why not? We need better organisms to cultivate. So amongst the blue mussel species, and I'm going to focus for a minute on northern hemisphere mussels because we know a lot more about their relationships. We have a series of closely related species here. These are all morphologically not distinguishable. And I want to focus in particular on the Mediterranean mussel, Mytilus gallo-provincialis. Now, that name, gallo-provincialis, is quite long, 7 syllables at my last counting. And so I will probably refer to it as MG or gallo throughout the talk, just because that is rather a mouthful. Now, this species, gallo, is widely established worldwide as an invasive species. And it's so much so that it's made the distinction of getting on the 100 worst invasive species of the world list. But what I think is really interesting is that these other species that it's closely related to are not invasive at all. So why, you know, this one species is invasive, but these others are not. And I think that's quite interesting. And so it has gotten this ranking as being a really bad invasive species, primarily from displacing native taxa in the Northern Pacific and South Africa. But there are other parts of the world that it's been introduced, and we know less about what it does there. So one of the places where we don't really know what's happening is in our own backyard in Australia. So is this species present in Australia? Well, we actually have mixed evidence. We do have blue mussels in Australia, and maybe they're endemic or maybe they're invasive. So I'm going to walk you through the evidence for both scenarios. So in Australia, we have distinct mitochondrial haplotypes that are endemic to the Southern Hemisphere. And so this is a study that was published many years ago now that looked at mitochondrial cytochrome oxidase 1 and used this to make this nice phylogenetic tree. What you can see is there's a large clade of Northern Hemisphere mussels, and then this clade down here, which has several subclades, consists entirely of Southern Hemisphere mussels, and this very distinct clade here is Australian only mussels. So this really looks like we have a endemic mussel species living in Australia. We also have fossils that predate European contact namely through ancient Aboriginal middens. And so this also suggests that we have a native species that was here before European settlers inadvertently brought over a European species. And this species even actually has its own name, Mytilus planulatus. And if you like history the way I do, it's kind of cool having species with that Lamarck provided that species name. But on the other hand, we also have evidence for non-native species present in Australia. And so if you remember, I told you that this whole clade is northern hemisphere haplotypes. Well, many Australian haplotypes fall within this clade as well. And so that's kind of the signature of an introduced species, when you have haplotypes that are sort of fall in a different part of the world from the rest of the sequences that they're related to. And in fact, there are identical haplotypes that are found in the native range in Europe and in Australia. And so these sorts of mitotypes in red have been picked up by a number of different studies sampling a number of places in Australia. So we really have evidence for two different entities living in Australia. So in fact, we have two different names registered. If you pull up Atlas of Living Australia, for example, you will find both Planulatus and Galloprovincialis, and their ranges look amazingly similar. And that's really because we can't tell them apart. We don't even know if they're both here. Okay, so but the point is not to criticize ALA, which I love, but just to say that, you know, we have a problem here that we don't know what we have in terms of blue mussels. So in the first part of my talk, I was going to be leveraging genomics to try to resolve this history of Australian Mytilus and to ask if we have distinct cryptic taxa present here in Australia, which would match the mitochondrial DNA results. But then to ask, is that mitochondrial DNA a reliable indicator of taxonomy? It's nice to have simple markers. It's really nice if you can use something like mitochondrial DNA to identify what species you have. That's quite handy. I'm also going to look at whether there's been single or multiple introductions of European Galloprovincialis. If there's hybridization, these species in the northern hemisphere, we know that mussel species hybridize quite readily, so it would not be unexpected to find hybridization. And then if we have a native taxon, how long has this been isolated from the European or a non-native form? And in the second part of the talk, I'm going to focus more on evolutionary changes that accompany introduction. So the first is more of a history and figuring out who we have here in Australia. So we used transcriptomes to get at these questions, and we decided to use transcriptomes because we wanted to target protein coding genes. We're interested in selection, but also mussels have rather a horrible genome to work with. It's large, there's a lot of repetitive DNA, and so we really wanted to sort of cut out that background problem and maximize the coverage we would have with our next generation sequencing. And so we used an RNA-seq approach. It worked out great. But just to emphasize, I'm not talking about gene expression differences. I'm talking about the actual coding sequences. So we can pull out the coding sequences for genes and align them across individuals and across different species. And that is the data that I'm going to be using. So to figure out who's who in Australia, we sampled from 3 different locations. We got mussels from Sydney Harbour, we got mussels from Bateman's Bay, and then also Spring Bay in Tasmania. We also sampled from Northern Hemisphere type locations. These are locations where in the native range of Mytilus galloprovincialis, including both Mediterranean and Atlantic locations. And that's going to be important in a few minutes. So I'm just going to show you this whole PCA and I'm going to walk you through this. There's a whole lot of things going on here. And this is a PCA based on over 20,000 SNPs. And so the first thing I want you to see is that the samples from the native range, the samples from the Atlantic and the samples from the Mediterranean are quite different from each other. And so we actually have a split of taxa within the native range. And this is not too surprising. There's been evidence for this before. Okay, so that difference is going to be really important part of our story. But we can also see that on the other axis distinguishes these native range mussels between a population in Spring Bay from Tasmania. And so these are quite distinctive. And here we have evidence for something that is not found in the Northern Hemisphere. And then these Bateman's Bay and Sydney Harbor populations are somewhat intermediate. So PCA is a really great way to explore your data. And another good tool that we have in genetics is to use an admixture analysis. So in this figure, each bar represents an individual, and the color represents the proportion of ancestry that belongs to a different group. And so if we look again at the Atlantic and Mediterranean, this is the native range of Galla provensalis, we see that the Atlantic samples cluster distinctly from the Mediterranean samples. And so these two analyses are completely concurrent. If we then look at the Tasmanian mussels, you can see that they are also distinct, but they form their own group with nothing else mixed in there. But then these two intermediate populations of Sydney Harbor and Bateman's Bay, we see here that they contain individuals that have allegiances both to one of the European populations, along with allegiance to these Tasmanian mussels. And so what this really looks like is that we've had at least two introductions, and these introductions have come from separate source populations. So we've had an introduction of Atlantic type of European mussels into the Bateman's Bay region, and we can see this as the purple in the Bateman's Bay. And then we've also had introductions from the Mediterranean group of Galla provensalis into Sydney Harbor, and we see this as the red component of their genome. So we've had at least two introductions of European mussels, but we also have this very distinct Southern Hemisphere Australian form. And so I'm going to call this Planiolatus for now. That is the historical name for Australian mussels. And so I'm just going to refer to them as our Planiolatus mussels. So the great thing about doing all this sequencing and looking at transcriptomes is suddenly just from a single mitochondrial locus, you have basically the whole gene, the whole plasmid at your disposal. And so what we did was we took all the loci in the mitochondria and that we could, and aligned them to make a grand network. And this is just to show you that we get basically exactly the concordant results, not surprisingly, with the previous CO1 studies. So here we have this super divergent clade of mussels that are consistent with being Planiolatus from Tasmania, that's consistent with this group. And then we have this clade of Australian mussels that actually nests within this bigger European clade. And so we have very consistent results with mitochondrial DNA, and this big divergence, though, between mitotypes that are found within Australia. If we now look at the nuclear loci, the story is completely different, however. So just these are outgroups. You don't need to worry about them. What I want you to focus on is this big bush up here, where we see that our Planiolatus types fall within this very little bit of differentiation from the Atlantic and the Mediterranean Galla provensalis. Okay, and so this sort of situation is really common if we have recent divergence or introgression. So it's consistent with both explanations, or either explanation, in terms of considering the relationships between these different taxa. And so simply looking at a gene tree, we can't distinguish between these two processes. And so what we decided to do was turn towards approximate Bayesian computation as a way of trying to tease apart the relative influence of divergence and introgression. So this allows for us to run simulations based on coalescence and to look at our species' demographic history and try to understand the relative effect of divergence and introgression on these species relationships. And so the results I'm going to show you from the ABC, I just want to emphasize that we're only looking at our putatively pure Tasmanian mussels. So I'm not including any hybrid mussels from Sydney or Bateman's Bay in this analysis. We're only looking at the mussels from Tasmania, and then we're going to look at them both in relationship to the Mediterranean variety of Gallo and then also the Atlantic variety of Gallo. So we'll consider all those relationships. And so for those of you who are not familiar with approximate Bayesian computation, the basic idea is that you simulate genetic data under various demographic models, and I'll run through some examples of models in more detail in a second. And from those models, you generate various summary statistics about polymorphism and divergence, for example. Then with your empirical genetic data, so what you actually found, you also look at summary statistics and calculate polymorphism and divergence. And then you compare these two in order to identify the model that is the best fit to your observed data. And this is your highest probability model. So this is done in the Bayesian framework. And so we did this using a standard array of models for two populations. So these range from panmixia, so no difference between our two species under consideration, just free interbreeding, basically treating as one whole species as if there was no difference between them, a strict divergence model, a divergence with divergence with gene flow model. So this is having gene flow throughout their history of separation, an ancient gene flow with divergence. Then we have divergence with secondary contact. That would be a situation as if they've been separated, but then come back into contact, such as with European mussels coming with European settlers. And then sort of a combination of those different things. So these are pretty standard models. If anybody's interested in details, I'm happy to talk to you about it at some other point. And I was super lucky that one of my former PhD students, Din Matthias, got really into ABC analyses, and he became really good at that. And that has been a tremendous tool set for our whole research group to use. And he's a lovely, smart guy too. So we assessed these models using both simple rejection and a neural net approach. And again, remember, we interrogated them against both Atlantic and Mediterranean varieties of Gallo provensalis. So the really important thing to see is that this model here that has ancient gene flow, but then divergence between the two populations, or an isolation period of no gene flow, this was the best support of our models. This divergence only model was also fairly well supported. But the really important thing to look at is the models that were not supported, okay? And so we had absolutely no support for a panmixia model. So if we didn't have different taxa, that would be the model that would be supported. But we can reject that model completely. We can also reject this isolation migration model that had continuous gene flow between the taxa. So whichever of the models gets accepted in the end, with these ones being the highest support, there is a substantial period of divergence between the taxa that is a period of sustained lack of gene flow between them, such as European mussels being safe in Europe and the Australian mussels sitting down in Tasmania or other parts of Australia, and them not interacting at all for much of their history. So that's the important part, is that the divergence time is strongly supported. So we can actually estimate that time of divergence. So if we take our best supported model here, we can estimate that divergence time, and we make some assumptions about the generation time of mussels. And this allows us to say that these two taxa have been divergent for 100,000 years or more from each other. And so to me, this really is a strong support for these mussels the Planulatus being its own unique form. We definitely have an endemic taxon of blue mussels in Australia, which I think is pretty neat. So in this first part of the talk then, we definitely have distinct cryptic taxa, but rather than just having two, we actually have three, because we have two distinct introductions of the putatively invasive Gala provensalis. We find that mitochondrial DNA is not useful at all for taxonomic ID, which is a little bit sad, because it would be lovely to go out and just use mite DNA to genotype things and know what we have, but unfortunately that's not the case. We have had multiple introductions, and there is definitely hybridization with these intermixed populations in Sydney Harbor and Bateman's Bay, showing that very clearly. But nonetheless, we also have at least one distinct population in Tasmania with 100,000 years or more divergence. And this, I think, is a really great start to uncovering this study system. Obviously, our sample sizes are not very large, and this is the downside with using transcriptomes, is that it's pretty expensive per individual, and so you get a lot of information per individual, but you're not able to sample super comprehensively in terms of geography or numbers of individuals per population. And this is something that we're trying to looking to fix in the future, looking to genotype with fewer loci, but trying to get more comprehensive coverage. And we're particularly interested in knowing what's being farmed, so what's happening in the aquaculture situation. So I don't have the answers to that, but that's something I really do want to know the answers to. But what I am going to focus on now is how we can use these systems to look at rapid evolutionary change. And so how can we use repeated introductions as experiments in nature, or in the wild at any rate? And in particular, I want to know whether these introduced Gala populations have undergone parallel genetic differentiation relative to their native range genetic background. So do we have replication of evolution happening again and again? And then are there some loci that are more prone or resistant to introgression in these anthropogenic hybrid zones? So parallel evolution is simply when you have a repeated event. So when you have a similar set of circumstances and you have the same outcomes in terms of evolution from those similar events. And probably the most famous case study in parallel evolution comes from this really groundbreaking paper that probably most of you are familiar with, looking at stickleback fishes. So in sticklebacks, there are two main morphologies. There's this heavily armored marine form, and then in freshwater populations, there's this repeated loss of armor. And so what was really great about this study was that these authors were able to identify a key locus for controlling that difference in morphology. So they identified this EDA locus, and then they sampled populations of fishes, both from marine settings in red, and then paired freshwater situations in blue. And so when they went and sequenced the EDA locus, you can see that there's this really strong divergence between these two different ecosystems. So the marine into the freshwater is accompanied by this divergence at the EDA locus. Whereas other loci that they looked at are just kind of no particular structure. Maybe sort of geography is the biggest overarching structure happening there, but you don't get that association between the morphology or the environment and this particular locus. And so this study kind of set up a whole cottage industry of people who are looking for parallel evolution, particularly when you have repeated colonizations of similar environments. And a lot of those studies have looked at marine to freshwater transitions. That seems to be a very common study framework, not the only one, but there is growing evidence for parallel evolution when you have repeated colonization to the same environment. And so we were interested in asking, well, okay, that's fine on a sort of historical scale, but when we're looking at really short-term scale, such as that of introductions caused by humans, do we also get that parallel evolution? And so I've told you many times now, and you are very knowledgeable in the fact that in the native range of Galla provençalis, we have this genetic difference between the Mediterranean form and the Atlantic form. And so we have different introductions from those. And in Australia, as I just told you in the first part of the talk, we have Mediterranean ancestry in our Sydney mussels, whereas we have Atlantic Gallo ancestry in our Bateman's Bay mussels. But we also have other introduction points around the world. And so we also have introductions of mussels in South Africa, where there is no hybridization with other metallists. There's no other metallists to hybridize from. And this is one of those really super invasive bad situations, incidentally. And then we also have introductions of Gallo into California. There is a native metallist, but there's very minimal hybridization. And so we have these multiple instances of similar lineages being introduced in different parts of the world, sometimes with a receiving community of mussels, of congeners that they hybridize with, and sometimes with a receiving community that lacks individuals with which they might hybridize. And so we can use this as a framework for testing and looking for what happens in these parallel and different situations. So do we have parallel genetic differentiation relative to the native range genetic background, and are some loci more prone to introgression in these anthropogenic hybrid zones? Because those are questions I'm going to be looking at in the last part of this talk. So this is the same PCA that you saw before, but updated with samples that we have from South Africa and California. So it basically has exactly the same layout and relationships, but what you can see is that these South African individuals fall very closely with Gallo from the Atlantic native range. And then we also have Californian individuals that fall within the Mediterranean native range. And we can see in our admixture analysis that these South African individuals fall with the Atlantic and these Californians fall with the Mediterranean. And so this means that from the Atlantic, we have 2 introductions we can look at, and from the Mediterranean, we also have 2 introductions that we can look at. And we can compare and contrast what's happened in these somewhat unplanned experiments. And so I'm going to give you a framework for how we can test for parallel evolution by looking at joint distributions of population differentiation. And so we're going to be looking at FST, which is just a metric of differentiation between pairs of populations. So I'm going to be comparing native populations. So this is the European native, the Galla provençalis, against where it has been introduced. So introduction site 1, and then we're doing the same native against introduction site 2. And so just showing some hypothetical data, we might expect to find something kind of like this, where we see a variety of different levels of differentiation. So very highly differentiated in one introduction against the native range, but not so in the other. But overall, in this fake data set, we would see a positive correlation. And so that positive correlation would indicate that there's genetic parallelism, that loci that are differentiated in one introduced site relative to the native range are on average also differentiated in the independent introduction site from the native range. And this parallelism could be due to selection. So if you have similar selective forces in the 2 introduced ranges, but it also can be due to certain properties of the genome. So we actually expect that regions of low recombination in a genome to be more prone to differentiation. And if that's something that you're interested in having a conversation about later, that's something we can talk about later. And so we can test for the significance of those outliers. And we're going to be using 2 ways of doing this. One is just the highest 1 percentile, but then also based on a joint a distribution that was developed by taking genotypes from native the first introduced and the second introduced population and permuting them and coming up with a null distribution. And again, that's something if you're particularly interested, I'm happy to talk to you about. So what does the real data look like? So first I'm going to show you the results from the Atlantic mussels. And so we have here our comparison between the Atlantic Gallo and South Africa. So this is our non-hybridizing point of introduction. And then we have our Atlantic, the native range again, and Bateman's Bay. So this is our hybridizing population. And you can see that overall there's quite a good correlation. So R squared of 0.5, it's highly significant. And so here we have really good evidence for genetic parallelism. So the same sorts of loci are being affected in both instances of introduction in these independent points of introduction. We also see in this, with this data that we have a couple of really strong outlier loci. And so these are loci that are strong candidates for being under selection in some sort of way in the native range. And I'll be coming back to some of these loci later. Now, in contrast, if we look at the Mediterranean source lineage, so here we have our introduction site in California, non-hybridizing, and Sydney where it is hybridizing. We also get a really strong correlation of 0.5, but we don't have any of those notable outlier loci. So the effect is maybe not quite as strong, but overall we have that parallelism sort of at the genome-wide level. So we get similar results when we compare what happens when you take a group of mussels from one source location and put it into two different parts of the world. And then we repeat that again with the other source population. We look at what happens. And we get similarities between the introduced populations when they come from the same source. But now what if we look between sources? Do we get similar outcomes when we start with different genetic material and put it in different parts of the world? And here we actually see a very different answer. So this is comparing what happens in our Atlantic comparisons and our Mediterranean-derived comparisons. And you can see that the correlation, although significant, is much reduced relative to what we saw a minute ago with 0.5. So here we have, by 0.1, here we have about 0.2. And so we get different outcomes when we start with different source material. So we have parallelism when we start with the same source material, but we get different outcomes when we start with different source material. And so that source really matters in terms of what's going on in terms of the outcome. So we can also look across loci at levels of introgression. And so this is, again, I'm going to give you kind of the setup in terms of what we're looking at, and before I show you some actual data, because it's a little bit of a complicated framework. And so what we're going to be looking at on the x-axis is, again, FST values, but looking at allopatric, Planulatus, and Galloprovensalis. Okay, and then we're going to look in the hybrid zone itself, and we're going to look on the y-axis, a metric of deviation of locus-specific allele deviations from the genome-wide admixture rate. So that's kind of a mouthful, but what it essentially means is a particular locus sort of, is the allele from at a particular locus moving on to the genetic background of other species more than you would expect. So loci that show strong positive or negative values are kind of out of alignment with the rest of the genome. Okay, and so those would be loci that are showing strong introgression patterns different from the overall genetic background. So we expect on average for loci to be sitting around the 0 point, and that would be indicative of neutral introgression. So if we have an loci sitting in this top part of the graph, that would be indicative of invasive introgression. So the loci that come from Galloprovensalis spreading through hybrid mussels disproportionately, whereas if we have loci that sit in this part of the graph, that would be indicative of local alleles resisting that introgression from the non-native species. And this could be due to local adaptation, so the local alleles are better suited to that environment, or it could be linked to reproductive isolation in the sense that there's a block between the non-native alleles and the native alleles. And so let's just look at some hypothetical data. Again, if we see a positive relationship, this will indicate that the same loci that differentiate the species are those that are most affected by anomalous introgression. And in this hypothetical example, more subject to invasive introgression. But we might also find loci down in this part of the graph. These would be loci that are candidates for local adaptation or reproductive isolation. So let's look at the actual data. So this is, I'm going to start again with the Atlantic example. So this is our Bateman's Bay population of hybrids. And so we do see this positive relationship, right? And so it's modest but significant. And we see that these same loci that are in general well differentiated between the allopatric populations are those that are on average introgressing more. So the Gallo-provençalis alleles are moving into the Bateman Bay population at an anomalous rate on average. So I'm going to look at that. We're going to go back to this graph in a minute, but I want to take sort of a historical pause before we come back to this graph, because there's another piece of information that you need for context that I have not disclosed to you. And so I'm now going to disclose this other piece of history that will make these results, I think, maybe even more interesting and hopefully not more confusing. So we've said again and again that we have these two different forms of Gallo-provençalis, the Atlantic and the Mediterranean. But what I haven't told you is that there's actually a third player in the scene in the native range. And this is the species Mittalis edulis. And so Mittalis edulis actually hybridizes to some degree with Gallo-provençalis, and alleles from Mittalis edulis introgress into Atlantic Gallo-provençalis. So in fact, part of the reason that the Atlantic and Mediterranean forms of Gallo are distinct is because the Atlantic form actually has the alleles that come from edulis. And so it's this really complicated historical scenario, but that introgression is stopped by the Strait of Gibraltar, which is a natural barrier to dispersal, which is where we expect introgression to be hung up. So really it's this third player that actually drives the dynamics between the Atlantic and Mediterranean taxa. So they're genetically differentiated, they have partial reproductive isolation, and many of the outlier loci, if you were to compare divergence between these two species, many of these outliers are actually derived from Mittalis edulis, and they might be involved in reproductive isolation in the sense that the edulis alleles have penetrated the Atlantic Gallo, but then they're fixed there, and the Mediterranean alleles are not able to reproduce with Atlantic Gallo that have those edulis-derived alleles. So it's a really complicated historical scenario. So what does this have to do with Australian mussels? Well, when we look at the relationships using a grouping of 3, where we include this edulis outgroup, what you can see is, not surprisingly, our Atlantic individuals have a little bit of that edulis history, but so do our South African mussels. So that those introgressed edulis alleles were taken with the introduced mussels over to South Africa, but they were also taken in small part over to Bateman's Bay as well. And so we actually have these echoes of past hybridization events that have occurred long in the historical past of these species. And so this influx of edulis alleles actually shapes what's happening today in Australia. So this is the graph I showed you exactly before, no change. But what I'm going to do now is I'm going to color in green alleles that are likely to be derived, loci that we know are highly differentiated between the Atlantic and Mediterranean, and are candidates for edgeless introgression. So these are loci that we expect the edgeless influence to be strong. And so what we see colored in green is that that slope line is higher than across all loci, and also the intercept or the deviation is higher. So basically, on average, these loci that have this edgeless component to their history are more introgressive within our Batemans Bay population. So that's really interesting in terms of thinking about how this long ago event is actually shaping hybridization that's happening here today in Australia. What's also really interesting is these two loci down here, which are sort of the furthest in that deviation towards Planulatus ancestry. So they're sort of outliers in terms of looking at Planulatus ancestry. And so these two loci actually were two of the outliers that appeared earlier when we were looking at the comparison between Atlantic and South Africa, and this should say Atlantic and Batemans Bay. So these guys are really interesting in and of themselves. And I'm just going to show you, zoom in on what's happening with one of these loci, where we can graph the frequency of the edgeless allele. So we're looking at variable SNP positions along the gene, along the contig. And let's look at the most differentiated SNP just for a second. So you can see that the really high frequencies of the edgeless allele, well, it's defined by edgeless. In our Atlantic population, we have 80% of that edgeless allele, but what we see in the Mediterranean, and also importantly in our Batemans Bay population, is that that allele is absent. And so what's happening in Batemans Bay is the mussels are retaining their native allele that is not that edgeless type. So they're resisting the introgression of that external edgeless allele that has been carried with the Atlantic Galoprovensalis to Batemans Bay. And so we think that this is a candidate locus for reproductive isolation, and obviously something we're going to want to look at a lot more in detail as we move forward. So we can also look at Mediterranean, what happens when you take the Mediterranean lineage. And so here we're looking at the Sydney Harbor mussels, and actually there's nothing interesting going on relative to sort of introgression in general, but also these loci that might be affected by edgeless history. And this makes sense because our Mediterranean mussels don't have that edgeless part of their history. And so we wouldn't expect them to have kind of weird stuff happening with regards to those loci. So again, the outcomes matter. The outcomes matter in terms of what is the source of the introduced material, whether it's the Atlantic or the Mediterranean material driving hybridization. So then to conclude this look at selection in our hybrid zones and across introductions, we do find evidence for parallel genetic differentiation, but it's only for comparisons involving the same source lineage. So that same source lineage has a really large effect in what happens kind of downstream or later on in the history of these introduced populations. We do see that some loci are more prone or resistant to introgression, but again, that origin of introduction matters. In Batemans Bay, we have this sort of really complicated situation where we have an excess of gallo introgression kind of at the genome level, but we also find some edgeless derived alleles that might be involved in reproductive isolation. So we kind of have all the answers going on there. In Sydney, introgression appears to be just sort of random and neutral, so not so interesting going on there. And if you want to read more, we're hoping to have that accepted very soon, but I'm happy to talk to you about it. So what does this all matter? I mean, I've been talking a lot about the history of mussels, which seems like a fairly arcane topic. But I think it's useful for us to think about these really closely diverged taxa. So when you have really recent divergence, you have taxa that are sitting in what's often referred to as the gray zone of speciation. So they're not clearly different species, but they are different from each other. And this comes back to this question of when do we value endemic diversity? At what point do we think that something is worthwhile in some sort of way? And so, you know, our Australian mussels are more divergent than a dog and the dingo. They're analogous to Neanderthals and humans. And, you know, so if we consider Neanderthals and humans different species, do we want to also consider Australian native mussels and this introduced Gala provençalis as different? In which case, you know, should we be referring to this as Planiolatus, or maybe give it a subspecies designation of Middleus Gala provençalis? Planiolatus, which is even more syllables, would be horrible. So I'm really interested in people's opinions. There are no easy answers on this, which is what I think makes this system really interesting. But names matter, and we know this from what's going on with dingoes. Whether or not dingoes have their own name or not greatly affects how these endemic species or endemic taxa are managed in the landscape. And so, you know, we need to think about how do we call things, because it does have real world consequences for how we might manage them. And just to highlight this with mussels, there is huge confusion in terms of what we have with mussels in Australia. So I pulled this up from New South Wales DPI, and I just want to read the last sentence of this description. Although the blue mussel in Australia, Middleus Gala provençalis, is similar to and share the same scientific name with the one from Southern Europe, it is native to Australia and has been found in ancient Aboriginal middens. Well, that's not really the story, right? The story is that we actually have both the European mussel and something that's distinct, and we can't tell them apart. And if we're calling them the same thing, we're obscuring that ability to tell them apart in any sort of meaningful way or to do anything about it. So it matters from what we call things, but also I think it really matters in terms of what's going to happen to these species or taxa. And of course, we don't know the answer. We know that hybridization is going on. We know that introgression is going on. I definitely think that at least the two locations we've surveyed in detail of Bateman's Bay and Sydney, we can describe them as hybrid swarms with low sample numbers. So there's a little caveat there. But what's going to happen eventually? Are we going to have genetic extinction? Are we going to lose that endemic Australian mussel? Is it going to be overrun by non-native alleles? Or are we going to have an enhanced invasive spread? You know, sometimes when you put differentiated things together, you get new combinations of genes that can act in different ways. And with Gala provençalis, I think this is not just idle speculation. So again, we know very little about Southern Hemisphere mussels, but we know in the Northern Hemisphere that the thermal range of Gala provençalis is considerably higher than other Middleus. So this is the mean sea surface temperature throughout its range, and you can see that they live at a much higher temperature waters compared to other Middleus species. None of these are invasive, by the way. And so several people have speculated that warming sea temperatures are going to give many invasive species, such as Gala provençalis, a competitive edge. And we know that Gala provençalis is adapted to warm temperatures. And so I think we actually should be a little bit concerned that we have, maybe more than a little, we should be interested in the fact that we have this warm adapted non-native species that has shown up in our coastlines and ecosystems. And so in closing, of course, I want to thank the person who really did all of the research, and this is Eva Popovic, who did a fantastic job with her PhD. I've basically been describing her PhD work, and I'm delighted she has stayed on as a postdoc working with me. She's now working on a completely different system with Sven Uthicki and Gert Warheide, and I'm sure we're going to have some really interesting results coming out of looking at crown of thorns. And as Katarina mentioned at the beginning of my talk, my group works on a variety of different species, and these are some of our other favorite species. And with that, I'm really happy to take any questions, and thanks for your attention.
Thank you so much, Cynthia. That was a very interesting talk. If anyone has questions, please, as I said, place them in the Q&A icon under your screen, on the bottom of your screen. I have two questions here, one from Jen. She asks, If I understand correctly, the Tasmanian population is the only endemic Southern Hemisphere species. I wonder how it got there. And do you think Mytilus was more broadly distributed than it is today, and it has become extinct from other locations in the Southern Hemisphere?
Okay, so those are great questions, Jan. So there are actually other Southern Hemisphere Mytilus species. It gets pretty complicated, so I kind of airbrushed over that part of the story. We have an endemic form probably in New Zealand. Whether or not it's the same taxon as Planiolatus remains unclear, but then there are also distinctive forms in Chile, for example. Question of how did they get here? You know, that's a really, really good question. To me, having a linkage, a natural linkage between Europe and Australia seems bizarre without it being more of a circumglobal type of relationship. I guess I would say the biogeography of southern mussels in general is not well resolved at this point, and that's something that I hope we or other people get around to doing, and maybe we'll have some more concrete answers to that. I mean, this is not the only example of antitropical species. So antitropicals are species that are sort of missing from the tropics, but are found in both northern and southern temperate waters. But it is really odd, I agree.
Thanks. Jen also says, thank you for the great talk.
Thanks, Jan.
I have a question. So you said that other Mytilus species were not considered invasive, but can you consider Edulis invasive as the alleles that are found in those populations are also found in populations in Australia?
Huh, that's an interesting question. I hadn't really thought about it in that way before. I guess my inclination is to say no. When I think about something that's invasive, it kind of has to meet two criteria. One is that it has been introduced associated with human activity. So it's moved beyond its historical native range. And then there's the more subjective part of the description. What is it doing once it gets there? Is it having a negative impact on the receiving ecosystem? And that's, of course, harder to answer. So with Edulis, the hybridization that occurs between Edulis and Galla provincialis within Europe is entirely natural. That predates humans traveling the world. And so I guess I would say that those Edulis alleles have sort of hitched a ride with Gallo that has moved to Australia. And so I'm not sure I'd put Edulis as invasive itself, but I suppose you could say that some of its loci are acting in a little bit of an invasive manner.
Thanks, Cynthia. I have a question from Ira. It says, great talk. Thank you, Cynthia. Can you comment on the range of the Batman's Bay and Sydney hybrids? Do you think they relate to specific introductions, for example, for certain aquaculture enterprises?
I would love to know the answer. I mean, this is the downside of doing transcriptome work, was that we were limited in how many individuals and how many locations we could sample from. And so I have a current PhD student who's working on answering that question with more comprehensive sampling. So stay posted and hopefully we'll have an answer on that.
Yeah, and related to these, do you think aquaculture plays a role in selection for these?
So that's another really good question. It could. I tend to think that if we have the non-native species in aquaculture, that's probably more accidental than anything else. In other parts of the world, in the Northern Hemisphere, aquaculture has actively introduced Galla provincialis. And so, you know, there has been a direct involvement of aquaculture. My understanding is that most farms are either using wild spat, so it's just they're sampling whatever happens to be in their location, or the few breeding arrangements that are going on are also taking mussels, adult mussels that they collect fairly locally, and spawning them and creating brood stock. So I think whatever we have happening in aquaculture here in Australia is probably just more symptomatic of whatever's happening outside of farms. Nonetheless, I think it would be really great if we could ensure that our farms are propagating the native taxon, and so that all the gametes that get produced by those farmed mussels are staying within the environment, and we're not unintentionally creating more gametes from non-native species.
Yeah, thank you. We've just run out of time, so it's 5 now. Thank you so much, Cynthia, for your great talk.
Thank you very much for inviting me, Katarina.
And thank you everyone for joining, and see you soon. Thanks.